just too many things. I'll record the just record the screen, and then I will OBR Studio, WebEx, I know everything. So okay, go on.
I'm gonna turn on my mic on this side. Assalamualaikum Wahid.
Okay.
Assalamualaikum everyone. So this is the paper that I'll be presenting. Stable depletion of RUNX1 ETO in Kasumi-1 cells induces expression and enhanced proteolytic activity of Cathepsin G and Neutrophil Elastase. So basically I chose this paper because my research involves siRNA, and that's a way that we use for transfection and for knockdown. So that's why they are doing stable depletion, which uses shRNA, as we will see, and they're trying to study the activity of this in different cell lysis conditions. So starting with the introduction, especially for Wa'id. So we are researching and they are researching on translocation Ta21 leukemia.
Sorry, Zen, can you make it bigger, please?
Yes. Can.
So what? Sorry, kind of slipped my mind. I'm sorry to interrupt you, Zen. Wa'id, can you hear us here? Can you hear?
Yeah, I hear you.
We cannot hear you.
I think my speaker. Can you say something, Wa'id?
No, I hear you.
Okay, good. So I'm sorry. Okay, it's just slipped my mind. Too many things. So this is the journal club. I believe this is your first time experience of going through a journal club session, right?
I think she can't hear us. You have to.
Okay, can you? Wa'id, do you hear me now?
Okay, it's clear now. Yeah.
We cannot hear you.
Your speaker is.
Oh, okay. Say again. Sorry.
I hear you now.
Okay,
Can you
good.
again?
Okay, so I'm sorry. I should have said this earlier, but I just had too many things. Okay. I believe this is your first time experiencing or going through a journal club session.
Yeah.
Okay, good. So in this journal club, we will select one of a paper that is related usually to our research. Okay, and then we will discuss, present and discuss. Usually we would have it in a PowerPoint format. We screenshot the results, what is the objective of the paper, and then the results, and then what do they find in their research, and then we discuss that paper in our group or in any groups. Okay. So that's the purpose of the journal club. Okay, so in this case today, Zain will be presenting the paper that he chose here, PlosOne. So PlosOne is, what is the impact factor for PlosOne? Okay, it's quite good actually. It's very high. Okay, so depending, not to say depending, but usually we would select a high impact journal or article to discuss. Okay.
Okay, okay.
Okay, so maybe next time it will be your turn. I mean, the first time it will be a bit difficult, but after you do it a couple of times, you watch your friends doing it, so you will be more familiar and you will be able to discuss that paper, inshallah. Okay, so here in this group, sorry, I also forgot to introduce. Sorry, yeah, it's just too many things. Okay, I'm going to turn my phone upside down. Okay, not going to reply all. Okay, so today in this group we have 1, 2, 3, 4, 5. Husam is not here. Amir is not well, so he's not joining the journal club session today. We have here Hanis. Hanis is the master's student. I should have asked you to introduce yourself, please. Sorry.
I think that's a good one. Hi, Assalamualaikum.
Waalaikumsalam.
Hi, Walid. My name is Anis. You can call me Anis. I'm a master student under Dr. Asmida. And my studies is about peptidomics in infectious disease.
Okay, nice to meet you.
Nice to meet you too.
And Zain?
Yeah.
Okay.
Assalamualaikum Wa'id.
Waalaikumsalam. That's it.
Yeah, we probably had a good chat before. My name is
Yeah.
Zain, and I'm from
Thank
Pakistan.
you.
No,
Thank
it's
you.
fine. It's fine, no worries. So yeah, I'm gonna be presenting Journal Club today because this is related to my study, which is about RUNX1 ETO leukemia, and we are also using the Kasumi-1 cell line. So inshallah, I will talk more about that in just a bit.
Inshallah.
Nice to meet you, Lau.
Good luck.
Thank you.
Ask her to introduce it to you.
Can you introduce yourself, please? Just a little bit about you.
Yeah, my name is Wadra Washte. I study medical laboratory science at Mu'ta University in Jordan, and I will, inshallah, study master's degree in UMS with us and Dr. Asmida. With Dr. Asmita. So it's the first time for يعني go to another country and contact with another people with another يعني that's nationality. It's not from our nationality, you know. And يعني I hope be a good يعني person for us and for my doctor. Okay.
Inshallah.
I'm
Thank you.
sorry.
Wait, no, that's fine. Thank you so much. Can you start, Doctor?
Yeah.
Anything else?
Anything to ask to? Okay, good.
All right.
Sorry, really. Sorry.
No, it's fine. So yeah, so this is the paper that I'll be presenting today. Stable depletion of RUNX1 ETO in Kasumi-1 cells induces expression and enhance proteolytic activity of Cathepsin G and Neutrophil Elastase. So just a fun fact for you, Wa'id. Olaf was actually Dr. Asmida's previous supervisor, so he also collaborated for this paper because he researches very specifically in RUNX1 ETO leukemia as well. So that's why now we are here at this moment in time. So RUNX1 ETO leukemia is basically caused by T821 translocation, and a little bit background in this is that RUNX1 ETO is found in 10 to 15% of AML cases. So the thing that we need to understand here is RUNX1 ETO acts as dominant negative inhibitor of RUNX1 dependent gene expression. So this is done by recruiting the core repressor proteins NCOR and SMRT bound to ETO moiety of the fusion protein. So this, I wish I had like a proper slide so I could show you guys the structure of RUNX1 ETO and how it works. So in this case, they are researching on specifically on Cathepsin G and IGF BB7. So this is because they want to see if the RUNX1 ETO, when you have a stable knockdown of RUNX1 ETO, how does that affect the let me show you guys. So the differential protein expression of target genes depending on the cell lysis conditions. And in this case, they were focusing on CTSG and the response to RUNX1 ETO knockdown, and they found that it was partially responsible for in vitro degradation of cellular proteins. So they use this phenomenon to actually see the using LCMS-based approach to see the proteolytic cleavage in RUNX1 ETO silenced Kasumi-1 cells. So that's why this was very nice for me, because they are also using LCMS as a tool to understand how the proteolytic cleavage happens, and we are looking at the metabolomic side of it. So it's pretty similar in terms of the tools, but not the same when you think about the application. So then they map the protease cleavage site. CTSG. That's Cathepsin G base.
Normal cell, what protein that you bought for what?
In normal cell, I think it has same function as in Kasumi-1, which is proliferation and differentiation. Yeah, and because it's like directly in line with RUNX1 ETO, so they were focusing on T specific, as far as I know. All right, so I won't go through the methods in detail too much. So they did the cell culture with the Kasumi-1 cells, and they did lentiviral transduction, and then they made the shRNA. shRNA basically produces stable knockdown, as opposed to siRNA, which I'm using, which causes a transient knockdown. So they use shRNA for the stable knockdown of RUNX1 ETO. Then they did the proliferation and clonogenic assay and flow cytometry for apoptosis and cell cycle analysis. This is something again that I'm also doing for the cell cycle using PI staining. And then MTS assay was used to understand the cytotoxicity, and then electron microscopy, Western blot, and Komasi staining. All of these methods we're also using in our research, and PCR. All of these, I will mention why they used it in just a bit. And then the sample preparation was something that was interesting for me, because they used the whole cell lysate in this, and they used RIPA buffer initially, and then they used the protease inhibitor cocktail. For us, the method that for MS analysis that we're using is MTBE method. So we get the polar and non-polar phases. And then they used all of this stuff, the LCMS data, with the statistics.
This is quite interesting, because I also scroll quickly, after you send the paper. I saw this RIPA buffer, I don't know if you never tried this method before.
No, because they basically lyse the whole cell with the RIPA buffer. Yeah.
So I was just thinking, while you were waiting for the result from
Panomics.
What do you call it? Panomics. So maybe you can just jump this to prepare this sample with RIPA buffer and send to Dr. Nazri and see.
We actually sent some samples yesterday to Dr. Nazri.
I know.
Yeah, but we use MTBE,
but
so
that's a mere sample, I know.
Can.
So I'm just thinking, perhaps this could make the difference. Maybe MTBE is not suitable for this cell line. So, and it seems quite simple.
Yeah, but I haven't seen this before in any paper actually. We saw so many, metabolite extraction methods, We tried so many also, but never saw REPA. I saw PBS, I saw water. REPA is for the first time. I think because they're trying to get the protein from the same
Proteomic analysis.
proteomic analysis, yeah. So it's not specific to metabolomics.
Maybe that's why.
That's why I didn't see this before. And even proteomics, I
Sorry,
think.
did they do metabolomic analysis or not?
No.
Okay.
They didn't do metabolomics.
This is
It's called
proteomic
protease.
analysis. That's
Yeah,
why they
protease.
REPA buffer.
Protease cleavage. But this was interesting for me because I haven't seen this before. That's what I was trying to say.
No,
What?
there are a lot of papers on the REPA buffer.
With metabolomics?
No. With the proteomics.
With proteomics.
Yeah.
Proteomics, I haven't read any papers. So I didn't know about this. Because I thought that REPA buffer is for lysis, and then we just use it for the for Western blot.
Yeah. I got confused there. Sorry, I got confused there.
In this case, we can use it for this also. So yeah, proteins were identified by false discovery of 0.01 on protein and peptide level. So this was like sort of a validation method that they used for this.
What is false discovery rate of 0.01?
I searched it up, but
Anis?
like it's like.
Can you hear me?
Waeed, can you hear Dr. Asmina?
Again, I cannot.
So do you hear us or not while we were talking here?
Dr. Asmina is asking, did you hear what we were talking about here?
Yeah, I hear what you are talking about shRNA. What? Yeah, I know.
No, the one that we are discussing just now.
So doctor asked me right now, what's the meaning of false discovery rate of 0.01?
Okay.
But I don't exactly remember. Like I know that this was using Swiss UniProt database, so they were trying to have a threshold.
Not necessarily. Sweet brought.
No, like they had like a database, and then they were trying to make sure that there weren't many false identification.
I saw it.
It's like 1% of the results declared as statistically significant are expected to be false or positive.
Okay.
So this is like the results are 99% correct. 1% is false.
That's another way to report your significance, apart from the p-value, you can also use the false discovery rate.
All right. And then they use a graph pad, a prism for the t-test and two-way ANOVA. All of these things I will show you guys the result in just a bit. So basically they started with the stable long-term RUNX1 ETO silencing in Kasumi-1 cells. So this was again with SHRE, which RE stands for RUNX1 ETO. And in this case, I'll just show you guys the figures. So if you guys see here, the control and SHRE, day 5, day 8, day 12, day 15. So they had consistent, if you see here the control, you can see RUNX1 ETO, you can see GAPDH is equal also. Maybe this one is not equal to this, but they had stable knockdown of RUNX1 ETO. And then they did the proliferation curve as well. So as you guys can see here that it starts from 0, all of them, and then control goes all the way up to more than 100%. But then SHRE actually stays around 40% of cell number. And then they did the colony formation assay as well. And so the colony numbers for SHRE were lesser than the control, which shows us that the SHRE was actually, it had an impact in the stemness of of the cell as well. And then they did the cell cycle distribution. So this showed us that the SHRE actually, when you knock down the RE from the cell, then that affects the G0, G1 phase of the cell cycle. And then they did the apoptosis assay, and they found that the SHRE cells were actually the I plus cells percentage. Yeah, so it had a reduced percentage of apoptotic Kasumi-1 SHRE cells in comparison to control cells. Reduced percentage. Sorry, I'm doing it this way because I didn't get time because of the workshop. And then, so after this,
Yeah, it's a common findings. Knockdown of RE won't induce much increase in the apoptosis, apoptotic cells.
I didn't do the apoptosis, yes, yet, but I did the proliferation assay, and the knockdown actually, like eventually, the SIRE cells finished by themselves after electroporation 3 by day 9.
Yeah, you would see that. You would have like a very flat.
Yeah, it didn't increase after that. And then after that, they actually, this part I had a little bit of issues with understanding because I haven't done this or I haven't researched more about this. So they did the testing with ABT 737 and doxorubicin. So if you guys see here,
ABT
I think
737
cells-
is also what do we call it?
it triggers mitochondrial apoptosis.
ABT. Oh, what's the number again?
737.
737. So it's the apoptotic inhibitor.
Yeah, that's what I don't understand, why they looked at the apoptotic cells and an apoptotic inhibitor.
It's a new drug. There is another isoform or another form of ABT. I just remember the number ABT 9 something. Venotoclax.
Oh,
Okay. It's not 737. Maybe 737 is a new form, but ABT 199. Yeah, it's venotoclax. Yeah.
venotoclax. Oh, so 737 is probably old then.
Not too old. 2017, 2018.
This paper is 19.
So it inhibits the BCL2 inhibitor. It inhibits the BCL2. Sorry, BCL2 involved in the apoptotic.
Apoptotic. Oh.
BCL2 is anti-apoptotic, so it's highly expressed in leukemia cells. So you have to inhibit the anti-apoptotic. Then it will increase the apoptosis.
Anti-apoptotic. So won't you knock down anti-apoptotic for inducing apoptosis? Like you would decrease anti-apoptotic.
Yeah, you can do the molecular mechanism by knocking it down, but you have to find a drug to.
Oh,
I mean, you can deliver the siRNA, but the therapeutic value of the siRNA is not like well established yet. So you design a drug that specifically inhibits that molecule or that BCL2 protein. Yeah. So yeah. BCL2 is increased in the leukemic cells. So that's why it's anti-apoptotic.
BCL2 is anti-apoptotic. So it doesn't allow the cell to go through apoptosis.
Yes.
Oh, makes sense. So it's like a survival mechanism for the cell.
Yes.
That's why it's
Understand now.
upregulated. Can.
So it prevents the cell death in the leukemia. So that's why even if you give the treatment and everything, but because of the high level of anti-apoptotic protein, so the cells are still alive. Understand? Yeah. Okay.
Yeah, so in this
Do you
case,
hear us the discussion?
I think you need to turn on your mic.
Do you hear the discussion, Wa'id?
Yeah, I listen. I hear you, sorry. We say the apoptosis and about proliferation that can low when you use shRNA. Yeah, that's mean.
Yeah, we are discussing. I will not expect any like right answer from you, because you're still new in this field. But at least you grasp the words or the meaning or the mechanism like what we are talking here. Good. If you cannot hear the discussion, please say something, okay?
Okay.
Yeah?
Yeah.
So
Sorry,
so you mute now? So what we discussed just now about ABT, can you go down? The ABT 737, okay? It's another form of it's another form of inhibitor for BCL2. BCL2 is anti-apoptotic protein. In leukemia cells, okay, the BCL2 protein is high. So it prevents the leukemia cells from going through apoptosis. So that's why if you give the treatment, BCL2 is still high. So the leukemia cells are still, they are not dead, they are not killed. So they are alive leukemia cells, okay? So they designed this specific inhibitor, ABT, 737, to binds directly to the BCL2. So BCL2 protein is diminished or destroyed, then leukemia cells can go through apoptosis. So that's the discussion.
By ABT 737, yeah?
Yeah, there are many forms of that already these days, because last time the first one was ABT, what was that? ABT 199, or we call it, the commercial name of it is venetoclax. Now we have the different version. Maybe there are some resistant mechanism when they're using the ABT 199. So there are certain resistance survival mechanism in the leukemia cells. So they designed the new ABT 737. Sounds like Boeing, huh? Boeing 737. Okay? So that was the discussion.
Thank you.
Oh, sorry.
All right. So they basically checked the apoptotic cells here, and they saw that the SHRE, there was a decrease in the apoptotic cells in SHRE. So that's why they did this apoptotic. I think, as far as I'm understanding this, is that they wanted to see the effect on apoptosis from ABT 737 and doxorubicin to validate this experiment. So they found that there was it displayed equal sensitivity to the anthracycline doxorubicin and BH3 mimetic ABT 737, which triggers the mitochondrial apoptosis. So basically, it had equal sensitivity even if the RE is knocked down. After this, they use electron microscopy. This is again something that I haven't done before, so this was interesting, but also I had a hard time understanding this stuff. So basically, the conclusion that they gave from here was that they had dilated cisternae in the Kasumi-1 SHRE. So if you guys see here, these are the arrows that show the dilated cisternae. This one, this one, this one. So if you see SHRE has more in number and more dilated than the control.
So what does it indicate, this dilated cystine?
This is what I was researching on. I think, as far as I read here,
What identifies as endosteric by the original related to dilated parts of the SSS.
myeloid differentiation. Yeah,
ER stress.
ER stress.
Above, go up a bit. One more, two more.
This
Yes,
one.
ER dilation is recognized as a sign of ER stress. Okay.
Endoplasmic reticulum stress.
Okay.
So it basically this shows that the release of RUNX1 ETO suppression in Kasumi-1 has been shown to induce myeloid differentiation and may present a conceivable explanation for ER dilation. So myeloid differentiation induces myeloid differentiation and then causes ER dilation. So oh, okay. So yeah. Because if the endoplasmic reticulum is dilating, then that means the cell is going through differentiation in this case. Because just for like the control, usually in Kasumi-1 cells, it is sort of like stuck in one phase, so that's why it doesn't go through the myeloid differentiation. That's what leukemia is. And then when you knock down the RE, when you see the EAR dilation here, endoplasmic reticulum dilation, then that means it's actually going through differentiation in this case. Correct? So after this, they checked the levels of control day 5 and control SHRE. And then in this case, BCL2 that we mentioned before, the apoptotic, anti-apoptotic protein, CTSG was Cathepsin G, and PU1, RUNX1, STAT3, and STAT5. STAT3 and STAT5 are linked with STAT3, STAT5. Yeah, so STAT3 and STAT5 both were the proteases that they were trying to see in this case as well, because that's what they did in the next step, the LCMS of the peptide analysis. And in this case, when they tested the SHRE for all of these proteins, they saw that for control, if you see here, control day 5 and control day 15, and then you see for SHRE day 5 and day 15. So BCL2 and CTSG was like initially increased, and later on it was slightly increased as well. And then PU1, RUNX1, STAT3, and STAT5. So BCL2 was the apoptotic, anti-apoptotic effect. Cathepsin G, as I mentioned before, was in the differentiation, and PU1. So they had, as far as I can tell here, that they had an increase in, so they had an upregulation of BCL2, CTSG, and PU1. And then they wanted to see the differences when you do high salt lysis and boiling SDS lysis. This was because they were doing the proteome analysis later on, so they wanted to make sure that the lysis way that they're using doesn't degrade too many of the proteins. And when you see here for high salt lysis, when you do SHRE, RUNX1 ETO decreases, RUNX1 decreases, STAT5A, STAT3, CTSG, Also, CTSG has less effect here. So if you see here, it's cleavaged into 3 different isoforms for CTSG. And if you see here on the boiling SDA slices, the CTSG bands are actually thicker on this side. And gap DH, doctor, I had one question to you about this. If you see the gap DH levels here,
Yeah, it's not consistent.
isn't that weird?
That's why I was wondering as well. The RUNX1 usually there will not be change
In SHRE.
in SHRE.
But that's something I saw in my SIRE, that RUNX1 actually decreased a bit also.
A bit.
A bit, like it was faint.
Just check the antibodies whether it binds to the N terminal, which is shared, or whether it's bind to the C terminal. So if they share,
Then when you knock down,
then you would see the loss, right?
yeah, you would see loss in
But then
RE.
RUNX1 ETO should.
In my case, RUNX1 ETO was less, but RUNX1 was also less.
Much less or?
Significantly, I would say like 40% less intensity of the band.
Gap pH was normal?
Completely
Equal.
equal. This was in the same one that I showed you, the BFKP knockdown. The same plot had RUNX1. So this is why here I don't suspect this, but the gap DH levels are. I was skeptical about these. Because if you see, there's a pattern, right? Gap DH here is less, and then they show less
Did they
here.
mention or explain anything about the high salt lysis?
High salt lysis.
Whether it affects the
Gap DH.
protein level?
Protein level. I think they mentioned it. No, this is MSMS.
This is figure 1, right?
Yeah. This is figure 1 I. Figure 1 IH. Prepared in Western blot analysis showed a reduction in RUNX1 ETO at later times during post-transduction, while the mRNA levels remained stable. Contrast, non-RUNX1 ETO, RUNX1 showed a consistent reduction on both mRNA and protein levels.
Okay. But move on because I think it's some time to really read about that.
Yeah. All right. In this case, the one thing that we need to observe here is that stat 5A, stat 3A, CTSG, all of them were decreasing, and a CTSG also formed. If you see here, there are 3 bands, Waid and Hanis. These 3 bands show that there was a proteolytic cleavage happening in Cathepsin G. This was the basis of their whole paper, actually. So moving on, they looked at the mapping of the protease cleavage patterns in RUNX1 ETO silence Kasumi-1 cells by LCMS-based peptide analysis. So in this case, as I mentioned before, they use RIPA to lyse the cells and extract the protein for the proteome analysis, and then for the proteolytic cleavage. Here, if you see figure 2b, as they mentioned here, figure 2b. So when you see here the control and SHRE, the peptides, if you see here, the tryptic peptides and non-tryptic peptides, so they are reduced in SHRE. So there was a reduction in the proteome of SHRE due to RUNX1 ETO knockdown. Tryptic. Trypsin. So a tryptic peptide uh, would would be cleavaged by trypsin, and it would cleave only the lysine and the arginine uh, residues. And if you see here, the this is the Komasi blue staining that they showed here. And if you see the proteome, it's actually shifted downwards, which suggests that this is actually cleavaged, or this is actually reduced as well in this case. And then moving on,
I just don't understand why did they use this high salt lysis and boiling STS lysis to.
I think they were trying to test that if it causes too much loss in the protein, which are linked with RUNX1 ETO knockdown. So that's why they were checking STAT5, STAT3, like these proteins specifically, BCL2, GAPDH. They were seeing these because if they were reduced too much, then that would mean that they cannot really show how it all combines together, like how the proteins interact with each other. Because if the lysis method actually lysed or damaged the protein that they were looking for, that might cause effect in the whole mechanism. That's what I think.
So they chose the boiling.
I think after this, yeah, they chose boiling as the lysis. Because here there's a clear separation in TSG as well. But still the gap DH is, yeah, really weird. Because basically if the gap DH is not consistent, right, then for example here they load less, so this would of course be less. So this is why. Anyway, moving on, they mapped out the cleavage patterns in the cells, and they identified that there were 17,419 90 peptides, and 5767 and 3370 peptides were unique to the Kasumi-1 control and Kasumi-1 knockdown. And then this showed that so basically 7.4% of the peptides were actually non-tryptic, and this reflects a fourfold increase of proteolytic activity and in SHRE. So this means that the proteome of the SHRE is actually more cleavaged, and it has an increase in the proteolytic activity in SHRE. And then moving on, they also looked at this graph, which again, because ICE logo is something I haven't used before, so I didn't understand exactly what they were trying to do here. But as far as I can tell, they were trying to show how the amino acid residues are split apart between these samples. So P1, P1, P2, P3, P4. Yeah, so these were the amino acids that they were trying to show here. And then yeah, this was the part which was the most interesting. And from here, when they saw the proteome and how the captains K, L, and S, so this will be important for us moving forward. So they showed that there was an overrepresentation of E and D at P1 and P4 of cleavage sites. So as far as I can tell, these are the cleavage sites, and glutamate and aspartate were actually more cleavage than the other proteins. So then they wanted to narrow down the spectrum of proteases, because see, they showed before that there's a four times increase in SHRE. So they are trying to specify which protease is actually responsible for this enhanced proteolytic cleavage. So they examined the RUNX1 ETO dependent expression of elaine and CTSKLS, as I mentioned before, and they found here that actually when you knock down SHRE, elaine was the one which was overexpressed as compared to the other proteases. So this is why they went ahead and they looked at specifically elaine. So when you knock down SHRE, here day 5, day 8, day 12, and day 15, all of a sudden, elaine is actually increased. If you see here,
Sorry, what is elaine again?
elaine is a protease that is the one that they identified was responsible for the 4 times increased activity of the proteolytic cleavage in SHRE. So elaine was the one who was the most
So you expect elaine to
increase.
right?
So if you see here, it shows that day 5, day 8, and day 15. You can see here day 5 it's a faint, day 8 is more pronounced, day 12 is more pronounced, day 15 is the most. So again, like Gabby, it's inconsistent throughout the paper. Only this one is the only one consistent. So anyway, then they wanted to validate this result, and they wanted to see what the downstream proteins will be affected by it. So RUNX1 ETO was again decreased. All of a sudden, here they're seeing that RUNX1 is actually increased. Yeah. So that means that elaine is actually responsible for the proteolytic cleavage of RUNX1 ETO, but not for the RUNX1. STAT5A, P1, P22, and elaine here.
What day
Because
is this?
this is
C. Kasumi-1 control work.
days 5 and 8.
With elaine specific or control SIRN.
SI elaine.
So when they knock down SHRE, elaine goes up. Now they knock down together, elaine goes down.
Yeah.
So you would expect RUNX1 to
increase, but in this case it's SHRE. But SHRE will decrease the RUNX1 ETO. So
no, in this case they don't show direct binding of RUNX1 ETO to elaine. They show that when you knock down RUNX1 ETO, increase in the proteolytic activities
by elaine.
by elaine. So the indicator is elaine, but they didn't show the direct binding of that.
I think they mentioned before that there's RUNX1 ETO dependent expression of elaine.
Dependent expression, but didn't say direct
Binding.
They didn't show in this paper. Dependent expression, not direct
Not direct binding.
Yes. So knockdown RUNX1 ETO, increase elaine, proteolytic activities also increase. Let me turn on my. Okay. So knockdown RUNX1 ETO. Honey, is you okay? Sleepy. Knockdown RUNX1 ETO. So increase in the proteolytic activities, catalyze, or elaine is an enzyme or what? Protein, enzyme. So catalyzed by elaine, right? So when they subsequent to that, the RUNX1 also goes down. So when they knock down on top of the RUNX1 ETO knockdown, they knock down the elaine, RUNX1 goes up. So meaning that RUNX1 also is affected by proteolytic activities by elaine. You see here?
Yeah, because of RE.
So dependent also.
Okay.
Understand?
So
I mean, at least from this diagram.
That's what I think. Yeah, because RUNX1 is actually increased, and the gap.
You know, you compare here with the here.
Yeah, SHRE. Also.
They just knock down RE here and control here. They did not knock down the ELANE. So SHRE remains, ELANE remains high, and RUNX1 down. So meaning that ELANE also targets the RUNX1 here. You know, without, there is absence of RE, even RE. So ELANE is like always there.
And ELANE is increased when you knock down RE, so it's sort of like ELANE could be a mechanism for reducing the RUNX1, so the RE can competitively bind,
Could
and
be.
cancer can increase.
Could be.
That could be the mechanism, because it does competitively bind, right? Both of them. Okay. Any question, Wade?
Yeah, sure.
Any questions, Wade?
Can I see what you are talking about now?
Yeah.
You say ELANE, it's a protease that reduce RUNX1, yeah? That's I understand.
Yeah.
Okay.
That's good enough.
Yes.
So this is a Western blot. This is a technique that we will also do in the lab, inshallah, when you come. So this Western blot actually shows us, I think you might have studied that before also. It shows us the protein levels in the cells, and we can check the proteins from this Western blot. And this is the RT-PCR, RT-qPCR. So this is the real-time PCR that we do here, and this is for the mRNA expression. So both of these techniques we actually use in the lab also. So they showed here that the ELANE was actually, when you knock down ELANE, the RUNX1 actually comes back to its normal levels, and then okay, this is the last discussion part of the paper, and I think I missed out a lot of key findings from this paper, but the one finding that we were focused on was using the LCMS data, which was about ELANE actually, because I completely forgot where it is. Okay, the contribution of ELANE and Cathepsin G is confirmed by shRNA knockdown, and they show that the footprints of CTSG activity on Kasumi-1 derived peptides that we talked about earlier for the protease, and then the CTSG and ELANE are, in this case, ELANE are able to activate Cathepsin B, which in turn activates CTSL via CTSD. Again, this is all talking about the mechanism of action of the proteolytic cleavage that happens inside the Kasumi-1 knockdown cells.
They did not prove this mechanism yet, right?
I think they proved it up here somewhere. I'm sure they did, because this is all
figure 3, sorry.
Yeah, figure 3, this one. So this is why they were doing all the other proteins here, to see how they're interlinked together. So this was also one of the findings, silencing as a consequence. So this is them trying to justify why why protease cleavage is happening in this case, because silencing of RUNX1 ETO in TA21 leukemia cells has been shown to promote a granulocytic differentiation accompanied by the expression of lysosomal proteins. So this is why the 4x times increase that we saw in the protease cleavage was happening, because it is to promote the granulocytic differentiation. So yeah, I apologize in advance if I missed out any key findings in this paper, because it was very detailed. There were many, I think 2 or 3 more key findings about the mechanism of action, but I didn't exactly understand how the different proteins are interacting together to cause the protease cleavage. So,
Okay.
yeah.
I think you
Sorry.
I think you missed out to point out in the first place when they, after the figure 1, actually they did mention that the prolonged knockdown of the RUNX1 ETO leads to the change in the protein here. What's the protein here? You know, they did the knockdown or the measurement protein analysis until up to day 15. So they saw the reduction of those proteins, but has not been proven anywhere else before. Something that's quite interesting here, because yeah, I would agree with that. Prolonged knockdown, we don't really investigate into that yet, you know. Something maybe we could also propose.
The one that I'm doing now could be considered prolonged knockdown, right?
Of course,
As I already just keep knocking it down.
it's for proliferation, but you don't see the reduction in the protein, you know.
In this case, they use SSRE, so maybe a stable long-term knockdown would have a different effect from like SIRE, which would be transient, like consistently changing.
Alright. Okay. Any question?
Very comprehensive and
You see, like they have only 3 figures. You see the storyline in this paper?
Oh,
Your only 3 figures from this paper. Okay?
3 figures and then 5 on top, 8 on top actually.
What?
There were more figures actually.
No, we just mentioned as 3 figures.
Oh,
The result is different.
Oh, yeah.
The results of the figure is different, but the way you tell the story, the way you write the stories, okay? So there are only 3 main figures. Figure 1 is to show that prolonged knockdown of the RUNX1 ETO leads to what is it? The different kinetics of the protein or mRNA expression. Okay. Day up to day 15 here. Okay, second figure. So when they see that the different kinetics of the protein after a prolonged knockdown, so they map to the proteomic study. Okay, understand now? And then the third figure, once they see the different proteomics analysis, different protease activities, they find the most, what is it?
The sort of like the validation, right?
Yeah, no, the third figure is the validation. Yeah. Okay.
But what do you think about the GAP-DH levels again?
Yeah, I'm a bit confused. Maybe they have their own reasoning there. I don't know. That looks a bit odd.
And also if you see the pattern, like gap pH is always decreased in this lane, in the lane where it matters the most.
Day 5, right?
Day 5, yeah.
Day 5 and
Day
8.
5 and day 8.
Can you share the screen because it cannot,
Oh, sorry.
it's not following us. Yeah, the gap pH should be consistent throughout your Western blot experiment, you know, because it's the control, experiment control. It's the same with your real-time PCR. This is your internal
This is the
control.
only one that was in a straight line.
They can still put the result, or they can pass the result given that the gap pH.
That's why I was so confused with this.
If it's higher, that's fine. If the bands is more thicker, you know, more intense, it's fine. And you see the reduction is not, you know. But in this case, because you're knocking down RUNX1 ETO and then you want to prove that it's reduced, the loading control also reduced, so meaning that you did not load equal amount of protein or what. So I don't know, to be honest. These people is in the Hanover, they are quite a good lab, you know.
Yeah, that's why not just this one, all of these first ones, it's consistent that the gap pH is actually loaded less. So this means that the protein loaded in these lanes are actually less, which sort of sounds like, you know,
This is in 2019, you know, even before COVID. Accepted or received September 9. I was on summer holiday in Malaysia. Accepted November 15, so I was back in Newcastle. What?
Cute.
yeah, you can map out
Yeah.
your whole life based on the paper.
Published December 11. This is before COVID. I don't know. Hanover, Germany. Yeah. Don't know.
I think I learned a lot from this, like for example, dilated cisternae and all of this stuff, extra stuff.
Anything else to ask? Hanis? No? Wa'id?
No, there's not.
No.
Thank you.
Okay then. It's almost 1 hour now. It's already 1 hour. Okay lah, good lah.
I have to go for Salah.
Oh, 11.
Yeah.
Okay. Alright, Wa'id. Zain has to leave for Friday prayers. Okay, inshallah we will meet again in 2 weeks time for another journal club by who? I don't know. I don't remember. Amir perhaps. Okay. So hopefully you got something out of this journal club and take the keywords, like for example the apoptosis, the Western blotting, what do they do, so you learn on your own first and learn molecular biology, especially, you know, because your basic is on the what do we call it, MLT, medical laboratory. Okay, so it's a bit difficult for you to learn, but somehow if you keep learning every day, you will get. Okay. Alright then, inshallah. I'll see you soon.
Have a good day.
You
Bye
too.
bye.
Bye. Assalamualaikum.
Bye bye.
You off already?
Laptop is dying.
Okay, before you leave, hold on, okay? Give me like